Our day began at the Tropical Rainforest Conservation and Research Center, where it is located at Elmina Rainforest Knowledge Center in Shah Alam, Selangor. Before entering the sampling site, we received a briefing by the facilitators on the methods we would be using throughout the practical. With our equipment prepared, my team made our way to the designated sampling area. This was where we would apply quadrat sampling to investigate plant diversity within a defined area. To make the quadrat, we used raffia string to establish a 5x5 meter quadrat. By defining a standard sampling area, we were able to compare the biodiversity within our sampling site in a consistent and systematic way. Our first practical focused on analyzing the variation in leaf morphology through statistical analysis. During this activity, we collected measurements from a total of 30 leaf samples of the same species. Each leaf was measured based on its length and width, and these measurements were then used to calculate descriptive statistics, including the mean, variance, and standard deviation. These values allowed us to understand the variation in leaf characteristics among the samples. Besides analyzing the variation within the samples, we also investigated the relationship between the leaf length and the leaf width through correlation analysis. All 30 leaf samples from the 3 sets were plotted on a graph paper using a scattered plot. Based on the distribution of the data points, the graph showed a scattered pattern, suggesting a weak correlation between the 2 variables. This indicates that leaf length alone might not strongly determine leaf width, as other biological factors may contribute to differences in leaf morphology. The second practical shifted our focus from individual leaf characteristics to the wider plant community within an ecosystem. This activity introduced us to quadrat sampling, a method used to study the distribution and abundance of plant species within the selected area. Within our designated quadrat, we recorded the plant species present and collected information based on their occurrence. With guidance from facilitators, we identified the species found within our sampling area before organizing the data for further analysis. Based on our observations, a total of 15 plant species were recorded within our quadrat. Among these species, Pomecia pinnata, also known as kasai, showed the highest abundance with 4 individuals recorded and a density of 0.16 individuals per square meter. This suggests that the conditions within our sampling area may have been suitable for its growth and establishment. However, the distribution of plant species was not uniform, as some species were represented by only 1 or 2 individuals. This variation in species distribution may be influenced by several environmental factors, including sunlight availability, soil conditions, competition between plants, and seed dispersal patterns. In addition to the practical activities, we attended an introductory session on conservation conducted by the TRCRC team. The session emphasized the importance of protecting tropical rainforest ecosystems, preserving native plant species, and restoring degraded habitats. It reinforced how scientific research and community involvement contribute to long-term environmental conservation. Afterwards, we were given a tour of the TRCRC nursery, where we were introduced to the conservation efforts carried out by the center. During the tour, we learned how native tree species are propagated and cared for before being planted in restoration areas. This highlighted the important role that plant nurseries play in supporting rainforest rehabilitation and biodiversity conservation. We were also given the opportunity to participate in a marine conservation program as part of our field experience. This program allowed us to understand and explore environmental conservation from a different perspective, focusing not only on terrestrial ecosystems but also on aquatic environments. As part of the program, I also participated in a poster competition that aimed to promote environmental awareness. I created a digital poster with the message "Save Marine Life" to encourage greater appreciation for marine ecosystems and the importance of protecting them. Overall, this field trip has provided me valuable hands-on experience beyond from what I learned from the classroom. Through Practical 1, I gained a better understanding of statistical analysis and the relationship between biological variables. Practical 2 introduced me to ecological sampling methods and helped me appreciate how biodiversity can be studied through field observations. Beyond the practical sessions, the conservation programs also deepened my appreciation for the importance of protecting our natural ecosystems. This experience not only strengthened my understanding of ecological concepts, but also highlight the role each of us play in conserving biodiversity for future generations. Thank you for watching.